So this is our last review session on the Wednesday, September 16th, in preparation for the class exam one that we have and the lab practical one that we have now during week 4 scheduled. And during this session, I was hoping that we can go through a little bit of the key points of what the chapters on endocrine system and the blood contained. And in addition to that, I want us to, while we're going through those key points, we will actually go through every single question that can come up in the exam. So I want us to just feel comfortable that there's no curveballs. I'll give you a heads up when there's going to be a more trickier question that you can expect to come up, where I would possibly try to confuse students, or where students often get kind of a little bit sidetracked. So I'll also give a shout out of those. Hi, Janessa, thank you for joining us. You didn't miss anything. I was just giving my normal starting speech of what we're planning to do today. So let's jump ahead to the plan. So I just really want to quickly revisit that where are we in terms of the course, and then the focus is going to be on this week's module. What needs to be done? Where are we in terms of the reviewing the material? And then I'll just finish off with talking a little bit about tutoring and study groups, and there's plenty of time for questions and answers. I would prefer if you shout out any questions that might come up, just when they do come up, that don't wait around till the end, because a lot of the time, at least me, if I'm thinking of a question and wait till the end, I forget what it was. And also, if there's something that I explain that didn't quite make sense quite as much, it is easier when you just say that this is now the concept that's complicated for me, so I know to focus on that. So without further ado, just really quickly to have a look. In terms of the entire course is 16 weeks long, and then we have the finals week. And for the finals week, we're doing something that's not as laborious as it traditionally is to take a finals exam. So we'll do something that's a little bit easier, a little more fun. But if you look at these 16 weeks that we've got lined up for this course, by the end of this week, we have actually completed 1/4, so 25% of the entire course. And by the end of this week, you have also, from the coursework, that means everything else minus the finals week, you have completed almost 25% of the grade. So that's the reason why I wanted to divide the material into smaller sections. So not everything is riding on one single exam. So it's kind of a little bit more low stakes. And especially since we're starting, I want you to get familiar with the system. I want you to get familiar with how the exams, how the lab practicals and all that works. So I'm amazed how quickly time is going, and it seems that this semester is going to be hectic as well. So just to kind of give you a little bit of a chance to give yourself a pat on the back that you are far. You're one quarter in already. So that's not a bad place to be. And I think you should be very, very proud of yourself. The first couple of weeks, maybe the first quarter, is the hardest of every course always. Just because there's so much new stuff, there's so much learning that how does the teacher, how does the instructor deliver the content, how are the exams formatted, and so on and so on. So very big well done for all of you for sticking around this part. That's a sign that you're going to do really well. And I do appreciate all of you who also joined me here on this session today. There's no assignments this week. So all of these that are listed on the screen were last week's assignments, but there's a reason why I wanted to list them there. I've gone through all of your grades, and had a look, and you're, as a class, doing really, really well. However, I know that sometimes in life it's busy and we might miss an assignment, or we might not perform as well as we want with an assignment, or things like that. So I'm totally open and totally happy to reopen assignments for you if you want to have another go, or want to have that first go of getting those points for those assignments. In terms of logistics, because there's a lot going on, the easiest way to do that is that you message me with clearly that what week or what chapter and what assignment it is that you would like to have opened. If it's all of them, then you just let me know that all. And then I can go and individually open them for you. That's not because I don't want everyone to have an access to those. That's just so I know where I have to go to update your grades. So if there's any assignments that you want to give another go, do know that I'm all about you having every single one of those points that you can earn by doing the assignments. So just message me and we'll get you set up to the point that you can gather all of those scores that count towards the final score. And like you knew, the assignments, we do them at the Connect from the publishers, their platform. I kind of already went ahead. What we are doing this week instead is that we have two things that we need to do. We need to do the exam and we need to do the lab practical. And that's all we're doing this week. Both are due by the end of Sunday. I believe it's 1 minute before midnight. And the reason why it's 1 minute before midnight is that that's just the way our system has been set up. That's the easiest way for us to manage that. So don't leave it till Sunday 11 o'clock. You might be then stressing a stress that you don't have to stress. Or if there's technical issues that throw you out of an exam or throw you out of a practical, all you have to do is message me. And this week I'll follow the messages extra carefully so I can get those unlocked for you as soon as possible. I know that there's a couple of messages about other things, and I'll answer to those as well, but I'm especially keeping an eye if someone has technical issues with the test or the practical. So those I want to be able to respond immediately. So no homework for this week, just make sure to do the exam and do the lab practical. And you also see the points distribution there. So exam 100 points, lab practical is about half of those points. So that gives you an idea of how much is available in terms of the points, how much you can earn points from that. What I'd like to do next, though, is to go to the study guide, and you can find the study guide from our Connect course and under the module for week 4. And what you'll find that it might be useful to either print this study guide or download it, or if that's not your learning style, you can just simply take notes, or you can just simply listen if that's your learning style. So whatever works for you. And I'll upload this video as well to the week 4, so you have a chance to go back and look if you missed any answers. But I would totally recommend you giving me a shout out if something comes up while we're going through this review sheet. So is there anyone who's totally confused where to find the review sheet or study guide, or is confused what it is? Well, I'm not hearing anything, which is a good sign. So what I'm going to do, we're going to go through the chapter material. And by the time we're finished, I think that this study guide is 17 pages long. We will have covered every single question that can come up in the test. So they're hidden in there, or not even so much hidden, but it's not one-on-one the exam written on a paper. So we will discuss also things a little bit larger than just the exam questions. I want you to have a whole picture of how do things relate to each other. But remember, it's an open book exam, so you can have all of your materials available for you. If I was a student, I would probably keep that handy if I have completed that. I would keep my notes handy, anything else that you've done while we've been studying during the past couple of weeks, I would keep those handy. And I'll give you a shout out when there's something that's especially important that you might want to have on your table. But it's again down to the individual learning style. I believe there's 50 questions in the test, and some of them are multiple choice, some of them are true, false. There's a couple of matching ones as well, I think. And I maybe put like one or two where you type in the answer, but they're really, really simple. Individual question to do it probably takes you like 30 seconds. So it's not really like they would be hidden answers there. It's just a matter of working through those. So let's have a look of the study guide, and I'm going to try to move to another screen. You should be now able to see the study guide on the screen that I'm sharing. Is there anyone who is not able to see the study guide? It's a good sign. I'm not hearing anyone protesting. So what I want to do, we'll go through the material, but also do give me a shout out if there's anything where you are kind of confused at what was that concept or what am I meaning by that. And also if you know an answer or are kind of like, oh, I've totally got this aced, don't hesitate at any point to shout out the answer as well. So we started the entire course, and I think that the endocrine system as well, but more so the entire course, by kind of having a look that there's many different systems in the body. Human body is made of as a combination of many systems, and it depends a little bit about the way we approach this, that how many body systems do we classify. Some classify, for example, certain systems together, whereas some divide them into different ones. So the number might vary a little bit, but what I wanted to take away from this was that all of these systems are interlinked to each other, and we have two control systems in the body that control all of the other systems, that what do they do, when they do, and how they do what they're supposed to do. And these two control systems that we have are our nervous system, which you learned about at the end of BIO201, I believe, so AMP1. And then we have the endocrine system, which we learned at the very beginning of the BIO202 discourse. So these are the kind of overarching control systems that oversee all other body systems, that how they work and what they do and at what intensity and how long and so on. So endocrine system is connected with all other systems. There's not a single system that endocrine system wouldn't be linked with. And that was also when I said that it's a little bit hard chapter to cover, and just because of the fact that a lot of the time we don't get to go to the nitty-gritty detail, I just mentioned that, oh, this hormone does this. But then we learn at the later chapters about this particular organ, that how does that hormone actually do it? So a lot of this feels often for students, I find, a little bit vague, or a little bit like you're getting only half of the story. But the rest of the story will be covered by the time we're finished with this course. So I just wanted to ensure you that there is a full story coming up. So I listed here the two control systems of the body. So we had the endocrine system and we had the nervous system. They both play an important role in regulating all other systems. And a lot of the time they might be both working at the same time or for the same goal. But there's significant differences between these two. And we remember when we talked about the nervous system and how it communicates within the body, that was all about those action potentials. So those things that traveled within the nerve cells, within the neurons. And if you think of an action potential, it is very quick to travel. We're talking about time lengths that are less than a second. We're talking about microseconds and so on. So nervous impulses are very fast. However, once a nervous impulse has traveled, then it stops. So our body often, a lot of the time, looks for changes. If something's constant, we stop responding or stop noticing it. If you think of, for example, of putting your hands to really hot dishwater, initially it feels hot while you're washing the dishes. But after time, you kind of get used to it. Or same when you, for example, go on a winter's day out there for a hike, you might initially feel cold. Or say you go to a swim on a cold day to a cold lake, it might initially, or a pool, it might initially feel cold, but slowly you get adjusted to that. So you stop noticing a stimuli that's constant. So our body looks especially for these changes. So nervous system sends only short-lasting signals, so often they're very, very easily picked up and noticed. Once an action potential has gone, that message has stopped. Nervous system needs to send another signal for us to get that message again. That's different from endocrine system, and you remember our endocrine system was all about hormones. So because hormones travel in blood, whereas nervous impulses traveled within neurons, it takes longer time for the hormones to find where they are going, do the binding with the receptor, and trigger the response from the receptor that they want to trigger. So endocrine system is much slower to act. This can be both beneficial and negative. The benefit of that is that often these signals sent by endocrine system are much longer lasting. So we can talk about days, weeks, or even months that the hormonal signals remains around and acts. So unlike nervous system, which was very immediate and fast responses for a short amount of time only, endocrine system, it might take a little longer to act, but it also remains active a lot of the time for a much longer time than nervous system. So that's kind of the big difference between those two. And I think that that would be a very good question in the exam, just to get us kind of oriented that what's coming up and what do we need to know. So just the difference between these two. And if at any point you feel that you want something clarified or rephrased, just let me know. Otherwise, I'll just keep going. Well, the next section that I have, I just simply wanted to give a definition. And definitions are on these multiple choice exams kind of nice, that you have multiple different terms and you have multiple different definitions, and then you have to match that, which one of those terms matches with what definition. So that's something that you can think of that might come up. As an example here, the field of studying hormones, studying organs that produce these hormones, how these hormones travel, and how these hormones act, and also how these hormones are inactivated or removed. That all is known as a field of endocrinology. So study of hormones and everything that goes with it. So whenever you see that word endo, something, you can kind of a lot of time be thinking that, oh, maybe that's a signal that we're talking about hormones. Or endocrine especially. And endocrine, that refers to something internal a lot of the time as well. So endocrine system served many different functions, and I've just listed some examples here. These examples are simply things that came to my mind when I sat down and I was like, let's talk about hormones. What could hormones do? These are not the only things that hormones do, and these are not necessarily the most important. These are just examples to get us to think about what hormones are and where they are important. If you think, for example, reproduction, Well, we know that hormones play an important role of that. Until you're old enough, you cannot reproduce until certain hormones start to be produced. And then during reproduction, hormones drive us to do a lot of things. Did anyone have any questions? I saw a mic come on and off. There's no worries if that was a mistake, but feel free to shout out if you had a question. Okay, well, the other thing that's a good example, I think, is if we think of a newborn during the very early period of life of the newborn, first months, even first couple of years, the newborn grows a lot in proportion to its body size in a very, very short amount of time. And it's not just that the newborn grows physically, the newborn learns new things, develops abilities, learns to control new things, learns to sense new things, learns to regulate its body, learns that it's even an independent organism on its own. So there's a lot of growth and development happening during the early life of an individual after birth. During that time, also the hormones that regulate these growth events and development events are found in great quantities in an individual. And another time when I think that this is especially well illustrated is when we go through our puberty. And you can often think if you have siblings or if you have kids or just know someone who's going through the puberty, they're at times a little bit all over the place. And that's not always just because of them. It might be also because there's so much hormones that are being now released. Some of them are new hormones. The body is reacting in a new way. So we go through, for example, a growth spurt during the puberty. And again, we can tell that there's going to be this growth spurt from regulating the hormonal levels of, for example, growth hormones in the blood. So those are two examples that I could think of. Then I just really wanted to, usually when I have a blank space where you write something, that's an important term or that's an important concept. You'll notice that sometimes I've also given the answer for you, but I believe that if you write it out, you'll probably remember it better. That's just one learning style. That's not to say that everyone learns that way, but blood is clearly an important one that I want to highlight here. So hormones play a lot of functions in maintaining things, such as maintaining fluid balance or maintaining body pH. Different things that we are regulating and keeping constant by having these hormones within the blood. And again, you can think of, okay, hormones within the blood, that's probably going to take a longer time to regulate these, but it's going to also keep the effects longer time around in the body than just the nervous system. So maintenance of functions and regulation of functions. And final one that I had there was body defenses. So you'll notice when we get further on this chapter, we just mentioned, or next chapter, we just mentioned the white blood cells. But when we study the immune system further, you'll notice that immune system works completely within the cardiovascular system and lymphatic system. So these white blood cells that defend our body from any pathogens, any foreign invaders, they actually work within the blood. So hormones play a role in regulating those, triggering those, and then these travel with the hormones within the blood. So that's just, I guess, that the biggest message that I wanted to send. If we continue kind of the introductory part, what I thought would be important for us would be to understand the difference between endocrine glands, and these are the glands that we focus on this chapter, so glands that produce hormones, and any other kind of glands that we might have in the body. I know that exocrine glands are not really the topic of our chapter, but I think it's important that we can tell these two apart, And I think that this could come up also in your exam, that you're able to tell what's the difference between hormonal glands or glands that produce hormones and any other kinds of glands. Sorry about the meow, it's just my cat who's coming to check out that what are we doing at this hour chatting about. So let's start by talking about all these other glands than glands that regulate hormones. And I think that the best way to think of this is by thinking of examples. So you all know we have sweat glands within the skin, and I think that you've covered the skin earlier on in AMP1. We also know that we have, for example, tear glands. They have a duct that opens right to the eye, and we know that we have salivary glands. We've got 3 different salivary glands types that we know, and we'll discuss those when we talk about digestion. But what you'll notice, all of these 3 types as well have ducts. They open within the mouth. And I forgot to mention that, of course, sweat glands, they are somewhere, and there's a duct from there to that very surface area on the skin there. So what we'll see that these glands, they all act very locally. Their secretions don't travel everywhere. So wherever they are, they have some sort of a tube that takes the secretion out to the very local area where it will have effects. So we don't have a salivary gland on our palm and then a long duct that would travel to the mouth. That would not make sense in terms of the design. So when we compare these exocrine glands into endocrine glands, what we'll notice that endocrine glands don't have these ducts. All of the secretions of the endocrine glands, all of the hormones are released to the blood. And then these hormones travel with the blood to all parts of the body. So endocrine glands secrete their secretions so that they can reach any part of the body at all, while it's traveling with the bloodstream. And I've listed there some examples. Really, examples for this would be all the glands that we will discuss about in this chapter. So do we feel okay about this similarities, differences between endocrine, exocrine gland? I don't see anyone protesting, so I guess that we're doing okay. That would be just a big picture idea that I do want you to have an idea of. I think a very good question in an exam would be that I would give you examples of different kinds of glands and want you to pick out the exocrine glands or pick up the endocrine glands of that list. And usually my examples would be the examples that you've seen on these notes, for example. You know that instructors love when there's something of an oddity or something that's not quite normal or that makes an exception to the rule. And that's why, while we find that most of the time a gland is either endocrine or exocrine, it cannot be the both. It's one or another. There are few exceptions where a gland is both. And pancreas is a very good example of a gland that functions both as an exocrine gland and as an endocrine gland. And what pancreas does as an exocrine gland is that it produces digestive secretions. And those are known as pancreatic juice. And actually, I think that those are the most complete digestive secretion that's produced by any digestive organ in our digestive system. And we'll talk about those a lot more, a lot later in our course when we focus on the digestive system. But what you need to know is that exocrine gland, those secretions that are made from the pancreas as an exocrine gland, those are released through pancreatic duct, where the bile duct joins as well, and opening is to the first part of the small intestines, duodenum. You don't need to know that just yet, but you know that it gets released to the very local site, very close to where the pancreas is located. Well, the hormonal function of the pancreas is different, and this is something that we do want to know. We know that pancreas secretes these hormones that regulate blood sugar levels so that we don't end up having too much blood sugar or too much energy going around. We remember sugars get converted into energy in the body because body is greedy and it just burns it all off if there's no regulation. So we store some amount of blood sugars for later use. And when we need energy, some of those stored blood sugars, some of those stored sugars are released back to the bloodstream. So it's all this regulation process of how much blood sugars to have around that pancreas takes part of. And two hormones that you want to know that relate to this blood sugar level regulation are, of course, are insulin and glucagon. Usually I would ask the hormone when there's multiple hormones that's the most that kind of makes sense, that's easiest to comprehend. But every now and then there's multiple options and I end up asking the second option, not the most immediate or most obvious one. So we all know that insulin has to do with blood sugar level regulation. I would also know that glucagon has to do with blood sugar level regulation. And the reason why I'm highlighting that it plays an important role is because there's gonna be other hormones in this chapter that sound fairly similar as glucagon, but they do other things. So as an exam question, it would be a really good question that can you tell out of those the one that's secreted by pancreas? Can you tell out of those the one that's involved in blood sugar level regulation? So just heads up that glucagon would be a good hormone to know and remember that that's all to do with the pancreas and all to do with blood sugar level regulation. Let's have a look of what I have next. And what I have next is a kind of a diagram that you've seen before. You might remember, and I'm sorry that it's tilted on this version. I can turn it around, but then everything else is going to be upside or tilted to the wrong direction later. So bear with me as we just quickly, I'll show this. So you've seen this before, and you've seen this in the chapter where we talked about the endocrine system. So at that time, I gave you a blank version of that. And I said that this would be a really good handout to fill in because it kind of summarizes everything that you need to know about endocrine glands. So you can write down there the name of the gland, the hormones that they secrete, and what those hormones do, and any other information that you can think of. And you can find this handout on our week 2 module. And you'll find that the completed answers to that on our review session for the week 2. That should be available there as well. I knew that there was issues with one of the links and that's been fixed. So all of the videos should work now. I'm not going to go through this in this diagram. I'm going to go through them as written out in the rest of the review sheet. But it's the same content. I've just, on this diagram that you see, I've moved the information that we'll talk about later to this version. So there's really nothing new. If you are a person who learns with colors, use colors. If you're a person who learns by writing them down, write them down. You might even test yourself if you've done this before, that, how much of that can I remember? And fill in the blank one. And then what you didn't remember, fill in. If you're an auditory person, you might want to read it aloud for yourself. I had a student who, one side, okay, I can put that. So what I would do, let's rotate it then. I always have a new version, so it's a rotate view. Here we have, if you didn't have a chance to see this diagram before, I'll open it up now bigger here. So we can do a really quick review of that. So the first gland that I wanted you to know was actually here, this group of two glands actually, hypothalamus and pituitary gland. So you'll notice that hypothalamus is often referred as the master gland. And the term master gland refers to the fact that it kind of has a control over all other glands that exist. So it controls a lot of other endocrine glands. So that's why it's called master gland. And from hypothalamus, we have actually a physical connection to the pituitary gland. So pituitary gland is located right underneath the hypothalamus, and this connection is known as infundibulum. It's just kind of like a bridge-like structure that then connects the hypothalamus to the pituitary gland. And when we see the pituitary gland, we'll see that there's two lobes to that. There's a posterior lobe and there's an anterior lobe. And these two lobes are separate both anatomically and physiologically. So we'll start with the posterior lobe of the pituitary because that receives signals directly from the hypothalamus. And there's two hormones that you need to know from the posterior pituitary gland: oxytocin, which plays role in birth and lactation, and ADH, which plays role in urination in terms of bringing the urination down. So we produce less urine when anti-diuretic hormone is around. So anti-urination. So those are the two hormones you want to know from posterior pituitary. And we'll talk more about these hormones when we go through them on this review sheet. Anterior pituitary gland does not receive signals directly. It receives those signals through the posterior pituitary gland. So hypothalamus sends a signal to the posterior pituitary that then forwards a message to the anterior pituitary. And hormones that we find from anterior pituitary are much more numerous. So we have growth hormone, abbreviated as GH. TSH stands for thyroid stimulating hormone. So this hormone regulates thyroid, and we'll have a look at thyroid a little bit later. We also have adrenal hormone, and we'll talk about adrenal glands a little bit later. So adrenal glands are regulated by anterior pituitary, as is the thyroid gland. So anterior pituitary oversees these. Then we have a bundle of two, FSH and LH. These stands for follicle-stimulating hormone and luteinizing hormone. These are hormones of the female menstrual cycle and also play a role whether the menstrual cycle leads into the menstrual bleeding, where we shed the uterine wall lining that has thickened during the monthly cycle, and also the egg cell, or whether we salvage the egg cell and salvage the uterine wall lining as well. So these two hormones, we'll go through them in greater detail towards the very end of our course when we talk about female reproductive system. And final one that I wanted to mention in terms of the anterior pituitary was our prolactin. Well, there's one more hormone that I want to mention from the brain, and that's secreted by our pineal gland. Our pineal gland secretes a hormone called melatonin, and many people who might have issues with sleeping or sleep-wake patterns, or, for example, travel a lot, different time zones, and need to adjust their sleep patterns, they might have seen melatonin being sold as an over-the-counter product. Melatonin can be also purchased on prescription on stronger drugs, but its main function is to regulate the sleep-wake pattern. Let's continue downwards on our list here and have a look of the thyroid. Thyroid is located at the anterior neck, just below, so inferior to our voice box, inferior to the Adam's apple, and so on. And thyroid is I describe it as a butterfly-shaped structure. You'll notice it has two lobes, one on each side, and they're connected from the middle on a structure called isthmus. What thyroid does, it regulates metabolism. So if there's overactive thyroid or underactive thyroid. Both of these situations could cause issues for us when we're trying to deal with the consequences of that. So either there's too active metabolism or too slow metabolism. So weight gain or weight loss that's resulting from hormonal imbalances. Within the thyroid, we have something called parathyroid glands. These are numerous little tiny glands within the thyroid. Really, in practice, when you look at a structure of a thyroid, for example, when you've dissected an cadaver, you cannot really see them with a naked eye. You would need to probably do some histology or some pathology to be able to tell them apart. But they are there. There's more glands that's main function is to regulate calcium balance in the body. So calcium is found in all parts of the body. You'll notice that calcium is involved with a lot of things. So that's the main function of the parathyroid glands. There's other functions that I've listed there, and we'll see later on when we go through the written version of this review packet. But calcium regulation is the important one for parathyroid. Then a little lower, just above the heart, we'll find thymus. And thymus was a little weird one because we rarely see it on an adult. Thymus starts as a big gland in proportion to the body size on a newborn. And then as we age, it shrinks, gets smaller, gets replaced by adipose tissue and connective tissue. But it doesn't mean that the function of the thymus stops existing. Just thymus is not physically found that large anymore. Thymus's function is to produce T cells, and we'll talk about T cells later on. T cells are cells of the immune system, white blood cells, and we'll talk more about that on that chapter that relates to that. But you'll notice that T cells were named after the site where they are produced, so they are from thymus. There's other cells, we'll talk about B cells in terms of the immune system later on, and those cells are produced in bone marrow. So again, they're named after the production site. Let's continue down the list and look at kidneys as the next one. Kidneys' main function is to regulate blood pressure. And it does that by filtering blood. And if we have to bring down the blood pressure, we might want to produce urine. We might want to remove some of that liquid. If we want to bring up blood pressure, we stop the production of urine, or at least bring it down, so we are not removing that much fluids anymore from the cardiovascular system. Of course, kidneys also filter the blood, so we remove anything that shouldn't be within. On top of kidneys, on both kidneys, we'll see that we've got adrenal glands. And adrenal glands are these small pyramid-shaped structures that are located on top of the kidney. And within an adrenal gland, there's two parts, cortex. Okay, kidney, but what does kidney do? I'll repeat that. So kidney's main function is to filter blood, and by doing that, regulate the blood pressure. So that's the big function of kidneys, blood pressure regulation. So a lot of the medications that are aimed for regulating blood pressure or aimed for dealing with too high of a blood pressure, so hypertension, they act on kidneys. So blood pressure regulation is the primary function. The secondary function that we find with the kidneys was that it does filter also any toxins out from the blood. So anything that shouldn't be there. Both kidneys and liver kind of clear your blood, remove what shouldn't be there. So for example, when you go for a night out on the town and drink alcohol, your kidneys and your liver are definitely not relaxing, although you might be. They're working very, very hard. And anything that shouldn't be urine for high blood pressure, just that kidneys regulate the blood pressure. And if we, for example, have too high blood pressure, we have hypertension, we might want to start removing fluid from the cardiovascular system. So we're removing liquid portion from the blood by kidneys. And when there's less fluid packed into small space, which is our closed vascular system, the pressure also drops. So if you have too high blood pressure and your kidneys want to respond to that, we simply remove some of the fluid from the liquid, or the liquid from the blood, fluid from the blood, and that is excreted from your body as a urine. So P is really regulation of the body fluids. And that too much fluids within the cardiovascular system correlates with the high blood pressure. If we have too low blood pressure, then kidneys become less active. We produce less urine. And within the urine, we also excrete anything that shouldn't be there, any toxins, anything else. Awesome. No, I'm glad you asked. That's exactly what I want you to do. Let's have a look at the adrenal gland. So adrenal glands, as you can see, these small structures on top of the kidneys, and you can see that the right kidney is lower. That's because the liver is located mainly on top of it. So it just kind of gets pushed down a little. But on top of both kidneys, we find adrenal glands. And adrenal glands have two different parts, similar to the brain when you remember from when we discussed nervous system on AP1, on BIO201, you'll notice that we had the cortex of the brain. Well, we have a cortex of the adrenal gland as well. And cortex was this outer surface of a structure. And then we have a structure called medulla. That's the inner part of the adrenal gland. And if we compare the cortex and medulla of the adrenal gland, we'll notice that cortex produces way more hormones than medulla. So there's a lot of hormones being produced from there. But I've grouped them into three that are the important ones that you need to know. First of all, mineralocorticoids. From the cortex, they regulate the electrolytes, the balance of the electrolytes. Then we have aldosterone. Aldosterone acts on the kidneys, energy production as part of the ATPase, regulation of the blood pressure, regulation of the plasma concentration, and regulation of the urine. So you see all of those functions that we just discussed in terms of the kidneys. So cortex of the adrenal gland regulates a lot of that. And the last group of hormones that I wanted to mention regarding the cortex of the adrenal gland was our glucocorticoids. Glucocorticoids are involved with the stress responses. And some people, when they're stressed, they might, for example, gain a lot of weight. And that's because these hormones also alter your metabolism. And glucose regulation as well. So that's what glucocorticoids do. You'll notice a lot of time that people are amazing to do in terms of being able to do things while they're under stress. But when that stress ends and the amount of stress hormones drops, their body might not do that well. They might health-wise collapse a little bit. That's because the stress hormones have kept them going. And then those stress hormones decrease and the body kind of has to catch up with what's been going on. The inner part of the adrenal gland has two hormones that we need to know from the medulla, epinephrine and norepinephrine. And epinephrine and norepinephrine were involved with the sympathetic branch of autonomic nervous system. You remember that this was the fight or flight responses. So when medulla of the adrenal gland releases its hormones, it has all to do with our body being prepared to either be in a combat state with someone, fight for our survival, or try to escape that situation. Both of these would mean that we, for example, direct our blood to large skeletal muscles, so we're able to run away or we're able to fight if we need. Our heart starts beating faster, stronger, pupils dilate, so we're able to observe as much as possible. And at the same time, certain other processes kind of go down. Digestion goes down. Urination. We don't need to worry about digesting or urinating if we're about to die. So that's kind of directing the resources where they're needed in terms of these fight or flight responses. And those were due to epinephrine and norepinephrine. Another one that's a good one here to notice is our pancreas. Our pancreas, we already talked a little bit about it, and the two hormones that I mentioned there as an exocrine gland and endocrine gland, or I mentioned two functions, the exocrine gland function and endocrine gland function. And what we want to know in terms of the hormones is the glucagon and insulin. Glucagon and insulin played a role in blood sugar regulation. Then I have bundle of two here. I have our ovaries. I don't know if I can show them at the same time. Let's go through separately. So we have our sex glands, glands that produce sex hormones. So we have our ovary and we have our testis. These are known as our gonads. And ovary, of course, the female hormones from the ovaries are estrogen and progesterone. Those are important hormones. And again, we'll discuss their functions in a greater detail later on when we have a look of the female reproductive cycle. But at this point, as long as you can connect estrogen and progesterone with the ovary. And then we have our testes on males, and these produce testosterone. And if we go back to the development of an individual, we'll notice that we are all initially set up so that we can become either one, male or a female. And during the embryonic development, it is the hormones within the body that then define that which one we end up being. And sometimes there can be issues with those, and we'll notice that there's oddities in terms of sex organs or sexual development, that we don't know which sex the newborn is. The last one that often gets overlooked is our leptin. And our leptin is secreted from the fat cells in our body, so adipose tissue. And leptin is a hormone that regulates our appetite. I think I misspelled the appetite there. Look past that and spell it correctly. You'll notice I'll never penalize you in an exam if you misspell things, because I misspell things all the time. Most of the time it's just because I'm a bad speller. Sometimes it is also because spelling might differ in the UK where I've been trained, but this is just me being a bad speller. So leptin regulates the appetite, and we can have clinical cases where one does not produce leptin, or this person doesn't respond to leptin. So in that case, the individual would have an appetite that never ends. They would just keep eating and eating and eating, and this can result in the huge overweight. They can become obese, morbidly obese. In lab animals, these lab animals without leptin or without receptors for leptin, they just eat themselves until they eat so much that they die. So this is an important hormone to know in that sense in clinical ways of managing these. So what we've done here, we've gone through the entire set of what I wanted us to know from the next few pages. So I'm not going to spend as much time on these pages then, since we already talked about those. This will be largely revision, so we'll go through them very quickly. Hypothalamus and pituitary gland, they came kind of as a bundle. So hypothalamus was the master gland that controlled other glands, and it was connected by a structure called infundibulum to the pituitary gland. And pituitary gland had both the posterior and anterior lobe. The posterior lobe received both neural, so direct neuron control from the hypothalamus, but also some endocrine control. And two hormones that you needed to know were oxytocin. Oxytocin plays an important role in childbirth. And childbirth is one of the few examples where we actually see the positive feedback in the homeostatic regulation mechanism in the body. And I have a diagram about the homeostasis later if you have forgotten what it is. But you remember in homeostasis, it's usually negative feedback that we see. We're stopping something from happening. But when it's positive feedback, we're promoting that thing that's happening to happen even more. So during childbirth, when the baby is about to be born, the head is pushing to the uterus and the uterine wall lining is being pushed, stretched. The stretch receptors within the uterine wall lining send signals that get passed to the brain. And within the brain, we have a set point at how far can we stretch the uterus. And if it stretches further than that, what happens, because this is positive feedback, we actually promote that and release oxytocin, which means that the baby pushes even further, the uterine wall lining stretches even more, and eventually the baby comes out. So that's unlike a lot of what we see on negative feedback, where we try to stop something from happening. In this case, we make it happen even more when this positive feedback kicks in. So oxytocin plays a role in that. Oxytocin also plays a role in lactation. So when the newborn starts to suck on a nipple, that releases oxytocin, which then is responsible for release of the milk from the breast. And more stimulation we have there, more oxytocin is being released and more milk is being released. So again, that's a positive feedback. Antidiuretic hormone, it does what it says in the tin, urination and deurination. So amount of urination goes down. In terms of the anterior lobe, all of those signals to anterior lobe come from the posterior pituitary gland. So signals must first travel from the hypothalamus to the posterior pituitary gland and from there to the anterior lobe. The hormones that I mentioned regarding that were growth hormone, often abbreviated as GH, and then, for example, thyroid stimulating hormone, TSH, and adrenal gland hormone. Both of these hormones can increase the activity of the respective organ, whether the organ is thyroid or adrenal. I mentioned follicle-stimulating hormone, often abbreviated as FSH, and luteinizing hormone, often abbreviated as LH. They regulated the menstrual cycle in a female. And then we had prolactin. Prolactin, again, name is in the tin, lactin to do with lactation, and it promotes lactation. So if you think what would be the hardest question that could come up with that, really it would be mainly matching the hormones with the glands. But I could ask, and I think that this question is in the exam, there's true and false statements sometimes. What would you say about a statement where I say that lactation, production of the milk, and secretion of the milk is regulated by hormones from posterior lobe of the pituitary gland? Would that be true or false? That would be false, because hormones to regulate the lactation come from both posterior and anterior. So you notice that true and false statements are sometimes not easier to do than multiple choice. You have to read all the parts of the question very carefully. A lot of the time in true and false statements, there's something that's true and then something that might not be true, or something that's true and another thing that's true. So I can first try to get you comfortable by telling that, for example, prolactin and oxytocin play a role in lactation regulation. both of them are being released from posterior lobe of the pituitary. That would not be true. The first part is true, that oxytocin and prolactin both play a role in lactation, but they are secreted from different parts of the pituitary. So that's kind of to give you an idea what sort of question you might expect. All of the other glands I listed then more condensedly. It looks like I've talked a lot about adrenal gland, but really not. It's just because it's divided on the cortex and medulla. And we discussed already the aldosterone in terms of the cortex, hormone from cortex, that has many functions. Really, the big thing there is that clinical implication, a condition called aldosteronism, where there's too much aldosterone. The other group that I mentioned from the cortex are glucocorticoids. This contains a group of various different hormones that play a role in regulating the metabolism, stress responses, and also glucose. Do you remember when I said from the pancreas that there was a hormone secreted from the pancreas that looked like something that you might confuse somewhere else? That was our glucagon. Glucagon is from pancreas, whereas glucocorticoids are from our adrenal gland and cortex of our adrenal. So that's kind of a disclaimer that just stay on the lookout for. The other thing that I do want you to know about cortex is that it secretes 25 different hormones. So a lot of different hormones are being secreted. You just needed to note these two, I think I highlighted. Medulla, instead of the adrenal gland, was involved with the sympathetic branch of autonomic nervous system fight or flight responses, and the hormones that you needed to know regarding that were epinephrine and norepinephrine, all to do with the fight or flight responses. And those we should have covered in our BIO201. Really at large, if you think of the adrenal gland, there's adrenaline, stress hormones, fight or flight responses. Let's have a look at the gonads. So gonads refer to ovaries and testes. That's kind of an overall umbrella term. And gonads secreted sex hormones. So those sex hormones we have for females from ovaries, estrogen and progesterone, and for males, testosterone. We really discussed a little bit about females and drug development and why we always use males in drug development. That's not a big concept that you have to worry about. I just listed it there because we did discuss it, but it's not something that's most important about this chapter to know. You know, whenever we talk about kidneys, we talk about renal structures. So you don't have a kidney doctor, you have a renal doctor. You don't have a kidney science, you have a renal science. So the word renal, just be familiar with that. You don't have a kidney artery, you have a renal artery. And the hormone that relates to the kidneys then is renin. So you kind of can remember it from the word renal. We have hormone called renin. Renin regulates the blood pressure. That was the main function. So that's the hormone you want to know, and the function of that is to regulate blood pressure. Then let's have a look at the rest of the hormones that we've already kind of gone through on that handout. We have our leptin secreted from adipose tissues, and the function of that was the appetite control. I showed an example on the lecture video about experiments that I've been involved in my research career, where we had rats from who we eliminated the production of leptin or receptors for leptin. And that caused these rats to just keep eating. And they became excessively obese until they ate so much that they died out of the amount of eating that they had done. We have some patients that we have had that cannot produce leptin or aren't responsive to leptin normally. I think the most common is that they cannot produce leptin. If this is the case, we can treat them by giving leptin as injections. And this helps them to control their appetite. So they actually can return to normal body mass rate and often recover from excessive obesity by being administered this leptin hormone, if that's the cause for their excessive obesity, that they don't have that hormone. So that's a very good one to know, even though it often doesn't get discussed as much as it should be. Pineal gland, we remember it was at the brain, actually at the third ventricle of the brain. Hormone secreted from there was melatonin, all to do with this regulation of sleep-wake cycle. I mentioned, for example, insomnia, or if you travel across multiple time zones, this might be a supplement that you might take to regulate your sleep. Pancreas, we already discussed. 2 hormones you wanted to know was insulin and glucagon, regulation of the blood sugar. I would want you to know about type 1 and type 2 diabetes. I don't know, is that a concept that everyone's comfortable and familiar with? I'll just do a quick review. So you remember type 1 diabetes is something that you're usually born with. And in type 1 diabetes, you either don't produce insulin or the amount of insulin that's being produced is too little. So as a result, for treatment of type 1 diabetes, so that we're able to regulate the blood sugar levels, a lot of the time there's injections that these individuals have to give, injections of insulin to themselves. Type 2 diabetes instead is often described as a late onset diabetes. That's something that comes later in your life, usually due to lifestyle issues, and this can be also managed to a great extent with changes in the lifestyle, what's being eaten and becoming more active, exercising more, and so on. So I could ask in an exam about diabetes and different types of diabetes, or one or another type of diabetes. We talked about parathyroid gland, those tiny dots, tiny glands within the thyroid gland. Their function was to produce parathyroid hormone, and this was all to do with calcium homeostasis. We also had thymus. Thymus was located kind of roughly over the heart, and the weird thing there was that it shrinked over the age. Thymus produced T cells that were important in the immune system. So T cells are named after the location where they're produced in thymus. Thyroid gland was located higher up at the anterior neck below your larynx. It had the 2 lobes and an isthmus that connected it. It produced hormones T3, which get converted into a hormone T4. And function of these hormones is to regulate metabolism. So I've listed there some examples like BMR, basal metabolic rate, tells us that how much energy we use just when we're at rest, not doing anything, just to keep the basic functions of the body going. Or we might need thyroid hormone. If we don't have thyroid hormone, we might have issues with the tissue growth or development of an individual, or blood pressure. So there's many examples of that. The one that I want you to know about the clinical importance with the thyroid gland is the Graves' disease. So know the signs and symptoms. So high metabolism, sweating, really high heart rate, really irregular heart rate, acting nervous, weight loss. All of these are signs and symptoms of Graves' disease. But that's as much as you need to know about that. But that could make a good exam question, that you can tell a little bit about what those signs and symptoms, to what condition are they related. So that was all you needed to know about the glands of the endocrine system. I did cover a lot later on this review sheet also about the basic theory, which gives you an indication that there's going to be questions about basic theory as well. Since we covered the handout about the glands, we're running a little bit behind in terms of time. If you have time, I will continue this review. However, if you have to go, you will find the rest of this review also as a recording. So I'm just putting it out there. Really, this first section that I have about the chemical messengers, this is a revision from our introductory bio 181 or bio 156. Really good one in terms of recognizing the terms hormones, autocrines, and paracrines. And then matching the definitions of these terms would be a good exercise in an exam. So we'll notice that hormones were these chemicals that are transported within the blood or within the lymph, and we'll talk about lymph later on, but they travel within the blood and they travel long distances everywhere in the body. Then we have another type of chemical messenger. This is something known as autocrine. And if you think of auto, you have automatic. That means that it acts on itself. Or autopilot. It pilots itself. So autocrines are chemicals that are secreted by one cell and they act on that very same cell. So it secretes chemicals that regulate the very same cell. So that's why it's called autocrine. Paracrines instead, there's something that's between hormones and autocrines. So they're secreted by a cell and they travel to cell or cells close by. Not all the way all around the body, but not on the same cell either. So very close by. I hope that makes sense. I would note those definitions so you can match them with the terms. You don't have to remember them word for word, but you can kind of tell which one goes with which one. There's different kinds of hormones, amino acid-based, steroids, and then the eicosanoids are the kind of one that we're still debating about. Really not much that you need to know about the chemical structure of hormones. I just added there because a lot of times students on intro classes are like, oh, steroids. Steroids are synthesized from cholesterol, so they're like, oh, should I eat very high cholesterol food? By no means, I don't mean that. That you get all the cholesterol you need from your body. You don't need to eat high cholesterol food to produce steroids. In terms of the hormone activity or hormone action, one of the good questions to ask when we talk about hormones, which is, I think, a logical question, which helps us to understand how hormones works, is that, okay, if we produce hormones and these hormones travel everywhere in the human body, why don't we then see effects of these hormones in every single cell in the human? Since they go everywhere. And since hormones are meant to trigger some sort of changes, why don't we see those changes in all parts? Why doesn't for example, our palm start to respond to thyroid stimulating hormone? Well, the reason is that although these hormones travel everywhere, for them to have an effect on a cell, this cell must have receptors. And these receptors must be a good fit with this hormone. So we talked about this key and lock mechanism. So they needed to be specific. So hormones and their receptors are very specific, highly specific. So only the hormone that completely matches with the receptor can trigger a response on only that cell. So we don't have receptors for thyroid stimulating hormone on our palm. That's why we don't see changes that you would think that that particular hormone would cause. I hope that that helps to kind of explain the importance of receptors. And what happens when a hormone binds to a receptor? Well, something in that target cell changes. It triggers some sort of a change. And I've listed there many examples of changes. As long as you know when a hormone that's a good fit with the receptor binds to that, some sort of a change happens within that cell. And this is where it's important, and this is an important concept, that we have two types of hormones. Some of the hormones are water-soluble, and some of them are lipid-soluble. And these water and lipid solubles should be a familiar concept from you when we talked about, for example, vitamins on introductory bio 181 or bio 156. If something is water soluble, it cannot mix with fats, with lipids, and it cannot get through plasma membrane, cell membrane, which is made of lipids. You remember plasma membrane was when the lipids organized spontaneously into two layers. So if it's a water-soluble hormone, it cannot pass the cell membrane and enter into the cell. These water-soluble hormones have to act on the outside of the cell. And how they act on the outside of the cell is that the receptors are on the cell surface. So when a hormone, that's water-soluble hormone, passes through the body and reaches to the receptor that's sitting on the top of sitting on the cell membrane, it cannot go through, but it finds that receptor. And this binding between the water-soluble hormone and a receptor for it, which is located on the surface of the cell, triggers that something else happens inside the cell. This is usually a message that's being passed somewhere else inside the cell, and then something happens. So we talk about second messenger when the message is not passed directly by that hormone, but we need another chemical to act as a transporter of the message or as a triggering the changes that will happen in the cell. Usually this is either activation of genes or altering the cell activity or something like that. So water-solubles stay outside the cell, tied to the receptors on the cell membrane, and need a second messenger to do their job. Lipid-soluble hormones instead are able to go through our cell membrane, are able to cross our plasma membrane. And because they're able to go through the plasma membrane, they can act on the receptors that are located inside the cell, within the cytoplasm, for example. And that way, they don't need a second messenger. They can directly go where they want to make an effect. And it can bind into a receptor inside the cell and there trigger whatever it wants to do. Often this is activation of certain genes or altering cell activity or whatever. So that's the big difference between those two. I would know that for the exam. I added a table there just because I had prepared it for you in your notes. If you feel confused or you want to know more about water-soluble and lipid-soluble, it tells you a little bit more about that. But really, what I've just told you, that's the level that I would expect you to do. Any questions about anything so far? Well, the next one should be just revision. So we've talked earlier on about homeostasis, and that's one of the key concepts in biology that will come up time and time again. Homeostasis is such a crucial concept that I want every one of my students to really know it by heart. And we see many different kinds of diagrams that explain what homeostasis does, but the diagram that I provided for you here is the most simple out of them. And that's why I use that, because then we can build on top of that diagram. So if you're thinking, what's the definition of homeostasis? Homeostasis was all about maintaining consistent internal environment. So it means that we're eliminating a lot of changes. We're keeping a status quo inside the cell. So not a lot is changing. We're keeping it consistent. Or in the entire organism. A good example of that would be, for example, body temperature. You don't want to get too cold and you don't want to get too hot. Because if you get too cold, you will probably die. If you get too hot, you will probably die. Too hot body temperature causes amino acids to start to break down, which then has consequences that are significant. It's not really that you roast yourself to death, but it's the destruction of the amino acids that starts about at 40 Celsius degrees, which is the concern for us. So that's why if your temperature keeps rising, you have to take medication that brings down your temperature, or you have to go to the hospital. It's not that you will just catch fire. It's that significant, important compounds in your body start getting destroyed because of too high of a temperature. If you get too cold, you can go to hypothermia. Your body systems slow down, and eventually you will become unconscious, and so on and so on. So there's the diagram that I provided about how the homeostasis works, homeostatic control mechanism. You notice there's something that changes, so some sort of a stimulus. And a good example of that would be the temperature. Let's say that we're getting now too cold. No, let's say that we're getting too hot. I used the example of too cold in another review session, so let's mix it up. We're getting too hot. Your body is starting to warm up too much. The room is super warm, and you know it's not good for you. So what happens? Your body has thermoreceptors, all the parts of your external body, but also inside your body. And these are sensing that your body temperature is now rising. And within the brain, which is the control center most of the time for us humans, there's a set point. We know that how high or how low your body temperature should be. If it's any higher, we're off from what we want to be. If it's any lower, we're off from what we want to be. You can think of that almost like as the control unit of the AC. So you set the range that is acceptable, and if it's anything else than that, then AC kicks in. Well, your brain has a similar set point for what the body temperature should be. And now in this example, it's getting hotter. Your sensor is telling to the brain that, hey, it's hotter than it should be. Well, we must react to it somehow. And that's where brain sends a message to effector. These are just various different kinds of ways how we can act against of whatever the stimulus is. So in this case, if we're getting too hot, what could we do? We could, for example, start to sweat. Our sweat glands would start to work, which helps us to have some of the fluids evaporating. And while we have fluids evaporating, they also remove some of the heat with them. Another example would be if we're getting too hot, we start to direct a lot of the blood to the skin surfaces. So again, the heat can be can evaporate, can be shared and can decrease with the external world. What else if you get too hot? Those are physiological responses. You could have also behavioral responses. You could go and turn on the fan, or you could go and have a cold shower. So those are all results of effectors. And the function of effectors is to stop this stimulus from happening. That's negative feedback. So a good example was that we're getting too hot. Now we're stopping ourselves from getting any hotter. So we're preventing the stimulus from happening. That's an example of negative feedback. There were a couple of examples where we can see positive feedback. And these were, for example, the childbirth, which we discussed earlier on. Blood clotting would be another example of positive feedback. And lactation, which we already discussed, was an example of positive feedback. But positive feedback in humans is much, or in life in general, is much more rare than negative feedback in the homeostatic control mechanism. I hope that that concept makes sense, because that is such an important one for us to know. Well, let's have a look here. I've just said that hormones can be released as a result of hormonal signals or nervous system signals. That's all I wanted to say there. Nothing bigger than that. Well, let's have a look. But I would know, like I said, the homeostatic mechanism. That's an important one. Let's have a look of a little bit. There's 3 ways or 3 factors that can affect to how strong of a response we're getting to a hormone from the receptor. First one of those is that how much of that hormone we have within the blood. Second one is that how much receptors we have for that hormone. And the third one is that how tightly these hormones and receptors bind together. So it's the balance of all of these 3 factors that define that how much of a response does a hormone generate. And these 2 terms I would know for the exam again, upregulation and downregulation. Well, an example of the upregulation is when we have very little hormone in the body circulating within the blood, but that's an important hormone and we want to know when that hormone is present there. So what can we do? Well, if we can't produce more hormone, if the hormone levels are always going to be low in the body, what we can do instead is that we produce more and more and more of these receptors for this hormone. So there's a greater chance that that hormone binds with the receptor, so we get a reaction to that. So since we're making more receptors, it's called upregulation. The number of receptors is going up. We're regulating that there's more receptors. If we have a lot of hormone in the body, we don't want to go crazy. And you remember, body was all about changes. Well, if we have constantly high amount of hormone in your body, we don't want to constantly trigger whatever that would trigger. What we want to do instead is that we want to bring down the effect of that hormone. And how we bring down the effect of that hormone is by removing some of the receptors. So we have only smaller amount of receptors left in the body for that hormone. So we're down regulating the number of receptors for that hormone. So those are two terms that I would know. What else have I told you? I have told you that you can find hormone both free in blood and bound to something in blood. And how we can control the concentration of hormone, that we make more hormone or we remove more hormone or less hormone. So it's a balance between how much hormone is being made and how much of it is being removed or inactivated. So this regulates the overall amount of hormone. There's another concept that I do want to mention, and that's called half-life. Half-life tells us how long time it takes for the amount of a hormone to be decreased by half. So some hormones have a half-life that can be days or even just hours. Some hormones have a half-life of months. So half-life is a number that tells us how long it takes for that hormone to be removed so that only half of the original amount of that hormone is left in the body. So I would know the definition, so I could match it with the right term. I would know how to write half-life as well. The last concept that you need to know about the endocrine system is these three definitions. Sometimes hormones need another hormone around, and this other hormone can have various different kinds of effects. I would know again these terms and definitions, so I would be able to match them. You don't need to be able to quote me the definition word for word. But I want that if I give you a definition, you can match it with that name of that hormone. So if it's permissiveness, this original hormone needs another hormone to be present so that it can actually act. So alone that hormone does nothing, but if there's another hormone, it can actually do its business. That's an interaction that's known as permissiveness. An interaction that's known as synergism is when we have the initial hormone and then another hormone comes. The initial hormone was already triggering an effect, but by adding that second hormone, it triggers even more of an effect, that initial hormone. So by having two hormones, we get a greater effect than by having just one hormone, even though there would be some sort of an effect. And the final example of interactions is antagonism. Antagonism is when we have the original hormone and then the second hormone comes, and by having that second hormone around, the original hormone cannot act. It stops the second hormone, from doing its function. So that's antagonism. So that's all you need to know about the endocrine system. And I realize that we've gone really over our scheduled plan and scheduled time. Again, if you still have time, we'll absolutely go through the rest. If you don't have time, again, this recording will be made available online during today. What we find, we had a chapter on the blood, and this chapter is kind of shorter to study because there's less material. On this chapter, what we have just introduction, that remember we're going to be talking about blood, but later on we'll talk also about the heart and blood vessels. Remember that blood was a type of connective tissue. Normally connective tissues are things like bone, ligaments, tendons. something very solid, something very strong. But in case of blood, it can be also liquid substance, but still blood connects different parts of the body. Blood is part of the internal transport system, so we're transporting many different materials with the blood. That's the main function of it. And blood is produced mainly in the red bone marrow, and the process of producing blood is known as hematopoiesis. I would know the names of these different processes of making something because it's always going to be poiesis. Just the word at the front will keep changing. So if we talk about making general blood, it's hematopoiesis. And we'll have a look at what other kinds of poiesis we have later on. The amount of blood in the body on an average person is about 8% of their body weight, which is about 5 liters, I think 1.3 gallons in an imperial system. I might be wrong. 1.3 fluid gallons, I guess. Something like that. In terms of the amount, just have a rough idea how much there is blood in terms of the percentage or in terms of the liters. The clinical relevance why we are talking about the blood is that analyzing the blood is one of the most simple and one of the most effective methods how we can study the health of an individual. What we do, we end up drawing a blood sample. That blood sample goes into a test tube, and then we put the test tube into a centrifuge that spins the blood sample at the fast speed. This fast speed causes the different elements of the blood to separate at different parts of the test tube. The heaviest elements go to the bottom of the test tube, the lightest elements go to the top of the test tube, and everything in the middle stays in the middle. So by doing this simple experiment, we can already tell the amount of plasma, the amount of red blood cells, and the amount of white blood cells and platelets in the blood. So just by doing that, and that's a very simple test that can be done even in a lot of third world countries and so on. Easy to do, cheap to do. We can tell if there's something obviously wrong with the amounts of these elements in the blood. If we have more resources, we can then have that sample sent to the lab where we can analyze that sample even more. And we can tell, for example, about the amount of hormones in that blood. And this gives us direct information of what's going on with the patient. So that's an important clinical relevance. The functions of the blood, I've grouped them into 3 different groups. Blood can play a role in transporting many different things, such as oxygen, carbon dioxide, nutrients, or waste products. So you'll notice that these are all the things that we need in cellular respiration or that are produced as a waste product of cellular respiration. So we're transporting all this in the body by blood. So into the cells that might need them and away from these cells that have produced them and away from body. Hormones were the other example that blood transported to all parts of the body. Second function group that I put was control of various different things. Blood plays a role in controlling, for example, the fluid balance. So we talked about the kidneys. If you have too much liquids in your body, the kidneys will start to remove some of that liquid and from the blood and make it into urine. If you are overheated, the blood can transport some of that heat to the surface of your body so it can evaporate. We can get rid of that heat. Or if you're too cold, we can then stop the circulation to the extremities of your body and actually keep the blood within the core and vital organs. So heart, brain, lungs, things like that that you absolutely need. If you're getting too cold, we won't be that worried if your ear or nose tip or fingers get frozen. You can live without them. But we really don't want to let those vital organs to get too cold and die. Another good that blood regulates is your pH. Look how narrow range for the blood we have in terms of the pH, 7.35 to 7.45. So really the acidity or how acid or how basic the blood is, that's regulated very, very carefully also by the blood transporting. The third and final group of functions that I listed for the blood was protective functions. So blood plays role in immunity, for example, transporting these immune system cells. Blood also plays role in blood clotting. So blood is able to stop us from bleeding to death. So that's an important function as well. So I hope you would know those functions. The final one on terms of that we want to spend time mainly on this blood chapter was to look at the different elements of blood. What is blood made of? And what do we need to know about these things? We already talked about spinning the blood sample and how we were able to separate these different elements then on that test tube. And the first element that we'll have a look of is plasma. Plasma is that kind of a yellowish liquid. It's a little bit thicker than water, and it comes to the very top of the test tube if we spin the blood sample. There's about 55% of the entire volume of the sample in plasma. So plasma is where everything else is dissolved to. So that's basically the matrix of that fluid of blood. Within plasma, we also have many different proteins. I've listed some examples there, and then we have a bunch of other stuff like nutrients, gases, hormones, waste, inorganic ions. But really, the plasma carries a lot of proteins. That's the important thing to remember from that. In addition to the plasma in blood, there's a lot of stuff that are dissolved within the plasma, but then get separated when we spin the sample. And these are known as formed elements of the blood. Well, let's have a look at the first example of formed elements, and that was our red blood cells. And the fancy name for the red blood cells is erythrocytes. So production of the red blood cells is again poiesis, in this case erythropoiesis. So recognize that whenever we have erythro, it refers to the red blood cells. So erythropoiesis refers to the production of the red blood cells. There's about 45% of the total volume of the blood is red blood cells. And if we think of the shape of the red blood cell, you remember it was like a ball that had been squashed from the middle. Not all the way through like a donut, but so that it's deeper from the middle. So we describe this shape as a biconcave disc. So it's kind of a flattened ball with the indentation in the middle. What we find from the red blood cells is that red blood cells don't have any nuclei, they don't have any cell organelles, such as mitochondria, in them. And I've told you before that all cells must have a nucleus. So how is this possible? Well, this is possible because at some stage of the development of the red blood cell, it has initially the nucleus, but at some stage of the development, that nucleus gets removed from the red blood cell. Same happens to the cell organelles. And this actually makes sense. If you think of the red blood cell, the main function of the red blood cell was to transport gases. So if we were transporting oxygen and we had mitochondria in a red blood cell, well, what does mitochondria do? It uses oxygen, and it uses sugars to produce energy, to produce ATP. So in that case, that mitochondria would probably start to eat away from that oxygen that we have on the red blood cell. And by the time the red blood cell would reach wherever it's trying to reach in the body, the oxygen might have been used already. So that wouldn't make any sense. So that's why we don't have cell organelles in the red blood cell. One thing that we do have in the red blood cell is hemoglobin, and hemoglobin is made of subunits, and there's space for 4 subunits of hemoglobin in the red blood cell. That's often abbreviated, the hemoglobin, as HB, capital H, lowercase b. And we talk about different kinds of hemoglobins depending on whether there's a lot of oxygen or no oxygen or very little oxygen bound to the hemoglobin of the red blood cell. If we have a lot of oxygen bound to the red blood cell, that red blood cell will actually look also different. That red blood cell, that blood will look kind of a bright red in color. So any blood that looks bright red is high in the amount of oxygen. Usually places where we get blood that's high in the amount of oxygen are places where the blood is coming directly from the lungs. So at that point, it's oxygenated in the lungs and it travels to the body rich in oxygen. However, a lot of our veins we see as darker than these bright red blood vessels. These darker, they're not really bluish if you look. They're kind of more of a purplish or dark purple in color. Textbooks often over-exaggerate that we have red and we have blue blood vessels. They're not really blue, but they are darker. They're more purplish. They're more bluish in color. These blood vessels carry blood where we have very little oxygen tied into the blood. So they're darker in color. Usually this blood is coming from various tissues where the oxygen has been already used and it's returning back to the heart. So the bright red red blood cells that have hemoglobin to them, these hemoglobin units are known as oxyhemoglobin. The red blood cells and the hemoglobin tied into those red blood cells where we have only very little oxygen, they're called deoxyhemoglobin. You remember D is opposite of something. Oxy refers, of course, to the amount of oxygen. So oxyhemoglobin, rich in oxygen, deoxyhemoglobin, poor in oxygen. So those would be two terms to know. What else we know? I mentioned that if we have a carbon monoxide poisoning, actually carbon monoxide binds to these hemoglobin subunits, and in this way stops oxygen from binding into them. So a lot of the gas poisonings that we have and deaths due to the gas poisonings are because of the carbon monoxide. Let's have a look at the red blood cell just a little more. We have situations where we might have too little red blood cells produced. In this case, we talk about hypoxia. If we have too many red blood cells produced, we end up with the blood being very high in viscosity. So it's all about this balance between how much red blood cells are produced and how much red blood cells are removed. Because red blood cells have about a lifespan of between 100 and 120 days, so you have to make some new red blood cells. And once red blood cells have been around for about 100, 120 days, they need to be removed. So it's always this balance. How much new is made, how much old ones are being taken away. That do we have the right balance of the red blood cells? What controls the production of red blood cells? Where there's a hormone, and since we've been talking so much about hormones, I recommend that you know this hormone. This hormone is abbreviated as EPO, and the name of the hormone is erythropoietin. Erythropoietin is a hormone that's secreted when there's too little red blood cells, and that hormone makes our body to produce more red blood cells. So normally your body responds to the loss of red blood cells by releasing erythropoietin. And as a response to the erythropoietin, you end up with more red blood cells. However, certain athletes have came up with a theory that if you have more red blood cells, you would be able to carry more oxygen to your muscles. And by having more oxygen in your muscles, you're able to work harder and you would outperform others in the sports. That theory has some truth to it, but not full truth to it. It's not just by having a lot of oxygen in your body that you're going to win a race. You do need to have exercise. You will need to have big muscles and so on and so on. It's not quite that black and white. But what we find is that some athletes want to go to the conditions that would cause them to produce a lot of erythropoietin, so they would end up with a lot of red blood cells. Well, what could you do to end up in a situation where you produce a lot of erythropoietin? One example would be to go to high altitude. When you're in high altitude, that high altitude causes you to to have less oxygen in your body. That's because there's less oxygen in the air in the high altitudes. So when you spend a lot of time in high altitude, your body starts to naturally produce erythropoietin, and you end up with more red blood cells to compensate for this lack of oxygen or this lower amount of oxygen. So that's allowed, and that's okay in athletics and in many different sports. So a lot of sports people go before an important competition to train somewhere in high altitude. That's why they have these training camps to all these great places like Alps and so on at the Rockies. And I'm always jealous where they go. Well, it's because they want more erythropoietin be produced so that they get more red blood cells. There is another option that's not allowed, that's considered as cheating in sports. You could inject yourself with a lot of erythropoietin, and that would cause red blood cells to be produced. So that's not allowed. I don't know, there's a little bit of an ethical issue that one could consider. Why is one allowed and another one isn't? I'll leave that for another time to consider that. I do want to mention that for you to be able to produce red blood cells, you do need to get some iron from your food. And then the red blood cells at the end of their lifetime are broken into various components. Heme, iron, and globin are examples of that. Did you have a question, Tanya?
Oh, yeah, I'm sorry. So, okay. So when you go into these high altitudes to produce this more red blood cells, and it triggers this hormone, why do you want to produce more like of the red, like what does that do for the athlete? Like I don't know, like why would you want it to do that?
That's a really good question. I'm glad that you asked that. So basically what you want to do, some athletes believe that they are only able to, for example, run so much, so far, because they ran out of oxygen, their muscles run out of oxygen. So they're coming up with a theory that if they were able to get more oxygen to their muscles, more oxygen to the various body parts that are acting, they would be able to run even faster or further. So that's why they want more red blood cells. I don't necessarily agree with that it would be this black and white, but it does give a little bit of a competitive advantage. The other example that we see is when you exercise hard, when you go, for example, for a marathon, eventually you have used so much oxygen that only oxygen in your blood that you're getting is, of course, from what you're breathing. And that's why you start to breathe faster and deeper when you're exercising. But there has been cases, because you then initially have less oxygen in your blood tied to the red blood cells, where some of the cyclists on these long cycling competitions have actually over time collected blood from themselves and put that blood into a freezer. And then during competition where no one's looking, they have stopped somewhere and tried to inject more blood into them. So they would do better. They would have more of these red blood cells because, again, of the idea that if you have red blood cells carry oxygen. So, okay, I have more oxygen in my muscles. I'm able to exercise even harder. Well, it turns out you can't do it like that. You can't collect your own blood and then inject it back to yourself. And not if you're not a medical professional and not if you're not working in a medical facility. These people actually, blood starts to clot when it's removed from human body. And in that blood that they're pumping back into themselves, there might be blood clots and they can get a stroke. They can get all sorts of pulmonary embolism. There's many things that they might end up with. So that was kind of a long-winded or taking the answer a little bit further, but to kind of recap what we discussed, the logic why they would want to train in high altitude or why they would want to inject themselves with erythropoietin is that by having a lot of erythropoietin in their body, either through injections or by spending time in high altitude, your body starts to produce erythropoietin. That causes you to have more red blood cells. So then when you return to the lower altitude, you have more red blood cells than your peers. Or when you have injected yourself with EPO, you have more red blood cells than your peers that you're competing against. And if you have more red blood cells' main function was to carry oxygen. Your body, your muscles, your cells are getting more oxygen than your competition. So if it's getting more oxygen, we remember that oxygen and sugars were what were needed to produce ATP, which was the energy that we used for everything in our body. So the logic is that then I'm able to produce more ATP, and ATP was used to produce or to drive various different functions in body, to be the energy currency in the body, that, oh, I'm able to perform better than my peers. I'm able to outperform my peers. Does that kind of make sense?
Yes, that makes sense. I work with a lot of like wrestlers and athletes, and I know that they go to the training center, which is in Colorado Springs. And I always wondered why they always go there before the competitions, or why the training center is there. I always wondered that why.
No, now you know, and you can kind of reason it that, oh, it's not just because we want to give them nice environment so they feel happy going to the competition. No, it's all about building up those red blood cells.
That makes sense. I knew it was the altitude, but I just didn't know what exactly. No one ever explained it to me like that before.
Well,
They just said the higher altitude. Okay, well, what does that mean? Like
that is like half of the explanation. Well, I'm so glad that that made sense for you, because that's the way you're going to remember things, when you can connect it into something that you know already.
Yeah, exactly.
I'm excited for you. You're going to do really well on this course, I can tell.
Oh yeah, and so I'm the one that keeps emailing you, because I keep finding like little issues or whatever when I take quizzes, or I get lost, or like earlier I couldn't log on. It said it needed a passcode, but then I had to like download this app, and like I don't know. I keep having like little things that I'm not familiar with. So actually,
So we're recording again, and the reason why I paused for a second, we just talked about with an individual student about a couple of things that related to that student's career goals and stuff. So I didn't want to have everyone listening to that. So we're back to where we were talking, and we were talking earlier on about the red blood cells, and I think we pretty much finished the portion on the red blood cells, but now comes the important part. This is the part that's going to be an extra credit question on your exam. So there's different kinds of disorders, and one of the disorders that you can get with the red blood cells is if you have low amount of oxygen in the blood. And you remember, red blood cells were responsible for carrying oxygen in the blood. So if you have a low amount of those, you have something that's known as anemia. And there's various different signs and symptoms for anemia. Anemic people are often very tired, very pale, have breathing problems, have chills, and all that sort of things. I, for example, am naturally anemic because my body doesn't absorb iron from the diet. So I have to take certain supplementary supplements to deal with that and so on. So if I didn't do that, I would look like a ghost walking around, and I would walk very slow as well because I'd be exhausted. So anemia is one of those clinical situations that can be caused by the lack of oxygen in the blood. But the reason why we have less oxygen in the blood is really the key for treating anemia. Just because we can diagnose an anemia, that doesn't tell us what to do. We can't just pump oxygen into the blood. We have to figure out the underlying reason why that patient has little oxygen in their blood. So one of the most common reasons for anemia is blood loss, that there's just bleeding somewhere. It can be internal, it can be external, but we're losing blood, and because we have so little blood left, there's not enough oxygen being transported. Other reason would be that we're not producing enough red blood cells, and that's, for example, in my case of anemia, because I'm not getting enough iron in my diet, I'm not being able to produce normally enough red blood cells. So because I don't have enough red blood cells, I have too little too little of the blood going around in body. And that's why I deal with it through a medical intervention. The other option, the third option would be that we are removing too much red blood cells from your blood. So red blood cells, which do get normally destroyed around day 100, 120, there's too many of those being taken away from. So it's the balance of these three things that can be one or combination of different things that causes anemia. Well, that's the foundation for what's really your extra credit question. Your extra credit question in the exam, which basically allows you to gain extra points. So for example, to the student who was wanting to really do well, even if you don't ace the rest of the exam, but you ace the extra credit question, you can still get so much points that you still go up. In my exams, it's usually always possible to get more than 100%. Sometimes students are a little confused. How could I get 120% in an exam? You can. It just carries that extra percentage to your overall grade, and then you can do really, really well. So the example that I'm asking in your extra credit question is a condition known as sickle cell anemia. And we had a look of the sickle cell anemia in one of the videos on this chapter about blood on week 3. Well, sickle cell anemia happens because there's just one single base difference in the DNA, one single base mutation in the DNA. Something's gone wrong when we've copied that DNA. And that one single base difference causes that we produce wrong amino acid when we're decoding that DNA code. And because there's one single wrong amino acid, the shape of the red blood cell is no longer this nice, round, from the center squashed red blood cell that we described as a biconcave disc. What happens instead, that this red blood cell becomes like a half-moon shaped, a sickled shape. It's not nice and round anymore. And because there's this shape change, the red blood cell is no longer able to fit in these 4 heme units inside it, these hemoglobin units. As a result, because there's no 4 hemoglobin units, it's not able to carry as much oxygen. And as a result of that, we now end up with the individual being anemic. So that's sickle cell anemia. The other problem that comes with not just not getting enough oxygen is that these wrong-shaped red blood cells, they get caught into the very smallest of blood vessels. They're not nice and round like the normal red blood cells that just slide through. These are a little bit jagged from the ends. They're the half-moon shape. They get caught. They're not quite as small. They can't squeeze through quite as small spaces as the normal red blood cells. So that can become an issue as well. So 2 issues, not carrying enough oxygen or getting stuck into small blood vessels. Well, generally we find that people from sub-Saharan Africa have usually got sickle cell disease. They're the most likely to have sickle cell disease. Not everyone by any means have, but higher percentage than anywhere else in the world of people with background from sub-Saharan Africa have sickle cell disease. It doesn't mean that you have to live there now. It means that your parents or your parents' parents or your more distant relatives were from sub-Saharan Africa. So we find sickle cell disease, for example, being more common in African Americans than other groups here in America. So that's not great for these people. But when we look at malaria in Africa, and we draw a map where malaria is common and how common it is, and we draw a map of sickle cell disease, we notice that these maps look almost similar. So wherever we find malaria, we also end up finding sickle cell disease. And that has made a lot of us wonder that why could this be the case? And a lot of research has been conducted on this. And we end up finding that malaria, of course, is not good for you, and how malaria parasite survives and reproduces is that at one stage of its life cycle, it has to enter similar to the human red blood cell or a red blood cell of another animal, but we're now interested about humans. The malaria parasite enters to the red blood cell and multiplies there and grows and then spreads further. Well, malaria parasite is able to do that with a normal red blood cell. However, with a sickle-shaped red blood cell, malaria parasite cannot enter in. It's wrong shaped. It's too sickled for the malaria parasite to go in. So as a result of this, people with sickle cell disease are protected from malaria. So we end up kind of wondering that, was sickle cell disease really a bad thing for these individuals? Maybe, yeah, they might not have quite as much oxygen in their body, but maybe it actually protects them from malaria. Whereas the rest of us would have to take malaria medication and so on and so on. So that's your extra credit essay topic. I will be asking you to explain what is sickle cell anemia, what are the consequences of sickle cell anemia, and how does sickle cell anemia relate to malaria. So kind of if you're able to tell that short story about sickle cell anemia, that's what you need to share with me and you can get the extra credit points. I'm not looking for general discussion of the anemia. You don't have to worry about that. I assume that you know what anemia is once you start talking. So just jump right in to discuss about the sickle cell anemia. So that's all about red blood cells. We're going to have a look of the white blood cells really quick. And white blood cells, the fancy word for them is leukocytes. So leuko is the word that we want to know with them. And I'm going to come back to this page in a second, but I want to roll down here and show that the production of white blood cells is known as leukopoiesis. So again, we're seeing this base of the word poiesis, making of something, and leuko now tells that it's making of white blood cells. So I would again know these different kinds of production of different cells, and I would be able to tell, is it red blood cell, white blood cell, or just general blood? So that's the introduction. We end up finding that there's very little white blood cells in the blood, probably less than 1% of the entire volume of the blood. And when we spin that blood sample, these white blood cells are located between the plasma and red blood cells. So kind of in the between of the test tube because of their weight. And there you'll end up seeing a very thin white layer. This white layer is known as puffy coat, and that contains our white blood cells and our platelets. So not a lot of white blood cells in the blood. But it doesn't mean that they wouldn't be important. White blood cells serve a purpose of defending us against diseases. And what's interesting about white blood cells is that when you get infected, when you get sick, when there's an invader in your body, some sort of a pathogen, the number of white blood cells grows then. So white blood cells are only about 1% in a normal healthy individual, but when you get sick, the amount of white blood cells increases. So we're dealing with the disease, so we're saying, hey, we need more soldiers, if you wish, to fight against that invader. So we recruit white blood cells and make white blood cells only when they're needed. So that's why, again, if you think of that, taking the blood sample, spinning it, and looking at the amounts of different elements, if we see a lot of white blood cells in that sample, we know that there's something going on with that individual's health. There might be some sort of infection going on or something else. So that's a clue for us. You can also get that information from a lab analysis of the blood sample, but you can also visually see that. There's 5 different kinds of white blood cells, and I think that Dr. Greer did a really good job of kind of showing what they all look like. And that all information is in our review sheet or study guide for the practical lab. So we're going to go through these white blood cell types. You will need to be able to identify them when you do your lab practical, but not in the exam. So exam, just focus on what I'm telling. Lab practical, focus on what's in your study. So let's go through these different kinds of white blood cells. And the first one that I want us to look at is neutrophils. Neutrophils are the most common white blood cell type. About between 50 to 70% of the white blood cells that we have in the blood are neutrophils. They have multiple lobes in the nucleus. They stay in duct. They have granules in the cytoplasm. Their function really is to protect you and help you to have immune defense responses against various things. The second one that I wanted to mention is eosinophils. Eosinophils are not quite as common. They're probably fourth common out of 5, so they're second least common type of white blood cell in the blood. About 2 to 4% of all the white blood cells in the normal healthy individual's blood. These have 2 lobes in their nucleus, so they are known as binucleate, and granules on their cytoplasm. Their function is especially to deal with parasites, deal with various different kinds of allergies. So that's the eosinophils. Next one that I want to have a look of is our basophils. Basophils are the least common of the white blood cells in a normal healthy individual's blood. Somewhere around 1% of all the white blood cells are basophils. They have, again, binucleate, 2 lobes in their nucleus. Their cytoplasm contains granules, and their function is mainly to deal with inflammatory responses and also the various allergies. So if you have an allergy, what allergy is a kind of a over-the-top reaction to something that should normally happen. We should normally protect ourselves from pathogens. Well, that's why we have developed the immune system responses. But sometimes our immune system is a little bit over the top. So when we're getting, for example, pollen in the air, our body goes like, oh, there's an invader in our body, I'm going to attack it. Even though we'd be fine just by accepting that, okay, there's some pollen in there. That is what allergies are essentially. Our immune system goes a little bit panicky over something that we shouldn't be that panicky of. So that's our basophils. And for that reason, if you need to manage our allergies, you can take antihistamines. Histamines are one of the chemicals that's associated with allergies naturally. If you take antihistamines, we remove that chemical and allergies don't happen. The next one that I want to have is the second most common type of white blood cell in the blood. Those are known as lymphocytes. So somewhere between 25 and 45% of the white blood cells in the blood are lymphocytes. Again, they have a nucleus. The nucleus is very large, spherical, takes almost all of the cell. They do not have granules in their cytoplasm, so we call them agranulocytes. Because the nucleus is so big, you can only see a kind of a small light blue circle on the rim of the cell. Their function is to deal with antibodies and immune responses, and really the most important ones of lymphocytes are T lymphocytes and B lymphocytes. Remember T cells. T cells were lymphocytes that were produced in the thymus. So they were named as T because of the site of production, and that was thymus. We do also have lymphocytes that are produced in the bone marrow. These are known as B lymphocytes because they're made at the bone marrow. And finally, the third most common type of white blood cell type, which was somewhere between 3 and 8%, kind of a kidney-shaped or horseshoe-shaped nucleus, cytoplasm, again, no granules. Function of monocytes is to eat pathogens. So anything that's not wanted in the body, these monocytes engulf it and break it apart, and then as small elements, they get rid of it. Well, what do you need to know about these white blood cell types? You don't need to know what they do or what they look for the exam. You need to know what they do and what they look for the lab practical. But what I want you to know for the exam of these white blood cells is the order, that what's the most common white blood cell and what's the least common white blood cell. Well, I will give you the names of the white blood cells. You just have to put them in the right order. And to make this easier for you, I have given you a nice mnemonic. The mnemonic is a sentence: never let monkeys eat bananas. If you look at the first letters of each word, that matches with the first letter of the white blood cell's name. So neutrophils, leukocytes, monocytes, eosinophils, and basophils. Neutrophils are the most common ones, and basophils are the least common ones. So if you're able to organize the white blood cells into that order, you're doing good for the exam. What else should we say about white blood cells? The very last thing that I can think of is to notice that overproduction of white blood cells is known as leukemia. Too small amount of white blood cells is known as leukopenia. So in leukemia, we have a lot of white blood cells around, and that's due to something that's normally not good news for us. And the final element in the blood was our platelets. About 1% of the blood were made of platelets, and the platelets are not actually full cells. They're only parts, fragments of cell that are present there, and they live for a very, very short life, only about 10 days each. So there's constantly platelets being produced. And the function of platelets is to cause blood to clot. And that's a good thing. So when you cut yourself and you slowly start to form a scab and that bleeding stops, that's thanks to platelets. Again, I can just mention as an example, I don't produce any platelets naturally. So if I don't take artificial platelets, or if I don't take any medication that causes me to produce platelets, I end up with just bleeding until I would die. So platelets are very, very important. And platelets are something that we need in that sense. That completes everything that I had about the blood in terms of the elements of the blood. And the very last concept that we've covered regarding the blood was blood transfusions and blood typing. What we noticed is that unlike what you have learned about Mendelian genetics, where you had two different genotypes, capital letter or lowercase letter for a characteristic, in blood typing, you actually end up with 3 genotypes. You can have a genotype where you have genotype that causes you to become blood type A. You can have a genotype that causes you to become a blood type B, or you can have a genotype that isn't A or B, so it doesn't carry any blood type other than blood type O. So blood type O means that there's nothing being carried as a genotype signal. And because you have 3 different alleles for these genotypes, you have the lowercase I, which coded for O. You had the capital I, subscript A, and then you had the subscript B on capital A. So you can produce either O, A, or B. Well, you remember that you get 1 allele from your mother, 1 allele from your father. So you will have 2 alleles for each characteristic. So the only way you can get blood type O, which meant that there's nothing carried in terms of the signal for what blood type that would be. And that would be having a lowercase I from both parents. That gives you a blood type O. If you have a blood type A, you've received either a lowercase from one parent and then the allele for blood type A from one parent. Or you might have inherited the blood type A from both parents. That gives you a blood type A. If you have blood type B, you have again inherited either lowercase i's from one parent and then the blood type A from one parent. Lowercase I from one parent and blood type B from one parent. Or you might have inherited blood type B from both parents, so BB. You can also have a mix where you have inherited blood type A from one parent and blood type B from another parent, which gives you a blood type AB. So we end up with 4 different blood groups: O, A, B, and AB. Well, who can give blood to who? I think that our video on this was pretty good, and I'm not going to ask in an exam for you to produce these tables, but I do think that being able to understand how these tables are done is kind of a good thing. What I am going to ask in the exam is for you to know who is a universal recipient. And if we look, the recipient AB, if you're blood type AB, you could receive blood from all other blood types. So blood type AB is known as our universal recipient because you can give someone who's blood type AB anyone's blood and they'll be fine. If you are blood type O, your blood can be donated to anyone at all, and the recipient of your blood will be absolutely fine. So that's why we call blood type O as a universal donor. That's kind of what I would want you to know about blood types. I have mentioned a couple of words about the Rh blood type. So when we talk about someone being positive or negative, that really is more of a problem if there's a mixing of child's and mother's bloods during the delivery of the child or the baby. And that doesn't cause issues immediately, but that will cause issues if the mother gets pregnant for the second time. So that's why usually the second pregnancy is much more carefully monitored in terms of pregnancy follow-ups and so on, doing the lab works. That concludes everything that I had about our study guide. I know it's been a lot. So what I have next, I'm going to jump back into the PowerPoint so we can wrap up. So we've gone through the study guide. What I want us to just notice is that I have also produced you a lab practical study guide. There it lists everything that you should see on those anatomy videos that Dr. Breer has kindly produced. So they are endocrine system anatomy and they are blood anatomy videos. So those are the structures. And your lab practical will simply be pictures. I think there's 25 pictures. And on those pictures, you need to identify something. And what you need to identify, they are all things that you have seen on those anatomy videos. So to prepare for the lab practical, if you want to look through those anatomy videos, this study guide shows you everything that you should see on each section of the video. There is also a practice lab practical on this week's module, week 4 module, that allows you to practice how will the actual lab practical that counts towards your final exam, how will it be? That's a good chance for you to practice and kind of get familiar with the format and feel comfortable that you know exactly what you're doing. It's also a good chance to revise a lot of these key concepts that might come up in the lab practice. So that's the link. You will see it on your Canvas shell. For this week, week 4, there is a little activity that says Lab Practical 1 Practice. It carries 0 points. So no matter how well or how poorly you do on that, it will not affect your overall grade. What will affect is the actual lab practical that carried 50 points. So just so you know that that's what you want to be able to do. So remember, if it's practice, that doesn't count to your final score. If it is instead, I'm trying to make this work. I'm not very good at this. If it is the lab practical, the actual one, that counted worth of 50 points. And the exam one, that counted worth of 100 points. So that's all I have to say about the lab practical. I have also produced the lecture slide handouts for all of the chapters that we've covered so far, and I will produce them for the later chapters as well, because someone asked, and I'm all about you ask, I will make sure that I get it done for you. And like I said, you have a chance to do these revision sessions that we have done. You have a chance to use tutors. You have a chance. I am happy to help you tutor you as well. Just message me. Or if you want to do the tutoring, we have the Cranium Cafe, and I have listed the details there. Do we have any questions at this stage about anything that we've covered? I guess that's a sign that we're feeling pretty comfortable. Like I said, I will upload this and the evening video from Monday to the canvas. If it's not already there, the evening video, I will definitely add it. So that gives you a chance to see that no one has been told something that you weren't told. Everyone gets told the same stuff. The evening and morning on Monday's video were exactly the same. So they will be available to give you revision as well. And I think we've achieved everything that I wanted to mention in terms of these objectives for today's session. And remember, all you have to do is the exam and the lab practical. No other homework for this week. And then next week, we will continue with our study of the cardiovascular system. So that's all that I have for this time, for this week's session. If you have any questions, I will be sticking around. Just let me know. But for now, I will stop the recording so that you can ask also questions outside the video.